Hello again after the break. We are going to start discussing the individual reactions of glycolysis one by one. In the first phase that I mentioned in the previous hour, two equivalents of ATP are used to convert glucose into fructose 1,6-biphosphate. And in the second phase, we can think that the fructose 1,6-phosphate, which is formed in the first phase, is going to be degraded into pyruvate with the production of 4 equivalents of ATP and 2 equivalents of nicotinamide and dinucleotide reducing equivalents. So we have to know that there are some intermediate steps. This is not happening at once, okay? There are some intermediate steps, and these intermediate steps are also very important. So let's look into the other steps. Here we can see glucose in the first step. Glucose is our starting molecule, and it is consists of 6 carbons. And we can see that there are some enzymes responsible to convert glucose into glucose 6-phosphate. These enzymes are hexokinase or glucokinase. Glucokinase is the enzyme which is present in the liver, whereas hexokinase is present in the other tissues other than liver. And we have to know that only one ATP is going to be converted into adenosine diphosphate. Then the glucose 6-phosphate is going to be converted into fructose 6-phosphate. We have got phosphohexose isomerase. This is another molecule. And the fructose 6-phosphate is going to be converted into fructose 1,6-biphosphate. And here we have got another important conversion. Another important enzyme is here, phosphofructokinase 1. And it is responsible to convert one. It is important to say here that one ATP molecule is converted into one adenosine diphosphate. Remember, in the first reaction, in the glucose to glucose 6-phosphate conversion, we lose one phosphate molecule. And also in this reaction, the fructose 6-phosphate conversion to fructose 1,6-biphosphate, we lose another phosphate. And the phosphate lost in these reactions are going to be transferred to the next molecule, whereas glucose is converted into glucose 6-phosphate. You can see that from the sixth unit of sixth carbon of the glucose, one phosphate molecule is added. And we can see another molecule, another phosphate is going to be transferred to fructose 6-phosphate, and that's why we can see this conversion takes place. So we have got an enzyme called aldolase. Aldolase enzyme is responsible to separate the separate the pathway into two. One is the glyceraldehyde 3-phosphate. This is 3-carbon molecule. Another 3-carbon molecule is dihydroxyacetone phosphate. The organisms should have to select one of them, and mostly we can see that glyceraldehyde 3-phosphate is chosen. So that's why mainly glyceraldehyde 3-phosphate molecule is chosen, because it has got a convenient skeleton to continue and to reach to pyruvate. So the glyceraldehyde 3-phosphate is chosen. We do not see the conversion of dihydroxyacetone phosphate. That's why the dihydroxyacetone phosphate should be converted into glyceraldehyde 3-phosphate. And we can see that 2 times 3-carbon glyceraldehyde 3-phosphate is formed. So we can see that the balance of the carbon, the balance of the number of carbon is kept constant. And the dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate by means of the triose phosphate isomerase. So this glyceraldehyde 3-phosphate is going to be our molecule to continue up to pyruvate. In this mechanism, we can see that glyceraldehyde 3-phosphate is going to be converted into 1,3-biphosphoglycerate, and we have got glyceraldehyde 3-phosphate dehydrogenase. This enzyme is also important. We can see that since dehydrogenase enzymes are present, we are going to think about nicotinamide dinucleotide is the most important molecule, and nicotinamide dinucleotide reducing form is the other most important molecule. We can see nicotinamide dinucleotide reducing form is formed, and after then, 1,3-biphosphogylacerate is formed. Of course, we can see 2 times glyceraldehyde triphosphate, 2 times 1,3-biphosphogylacerate. We have to multiply every molecule by 2 after glyceraldehyde triphosphate is formed. Because it's 3-carbon molecule and we have to keep the carbon number constant. 2 times 1,3-biphosphogylacerate is going to be converted into 3-phosphoglycerate. Here we obtain 2 ATPs as well. We see that 2 adenosine diphosphates are going to responsible to enter into the pathway, and we end up with 2 adenosine triphosphates. We can see phosphoglycerate kinase is formed. Phosphoglycerate kinase is the enzyme that helps to form 3-phosphoglycerate. So we have got another reaction. There seems to be a change in the phosphate location in the molecule. 3-phosphoglycerate is formed since one phosphate molecule is transferred to, or 2 phosphate molecules are transferred to 3-phosphoglycerate. And the orientation of the 3-phosphate which is present in 3-phosphoglycerate is changed, and we obtain 2-phosphoglycerate. And 2-phosphoglycerate molecule is then converted into enolase, and we form 2 times 2-phosphoenol pyruvate as well. We just come from 2 times 2-phosphoglycerate. By the help of enolase, we can see 2 times phosphoenol pyruvate is formed. So these 2 phosphoenol pyruvate molecules are able to be converted into 2 pyruvate molecules by the action of the pyruvate kinase, and this pyruvate kinase is able to show the conversion of 2 ADP molecules into 2 adenosine diphosphates. Let's summarize the reactions. The hexokinase reaction is the first step of glycolysis. So the adenosine triphosphate dependent phosphorylation of glucose to form the glucose 6-phosphate is the first reaction of glycolysis, and this is catalyzed by tissue-specific isoenzymes known as hexokinase. Isoenzymes are the enzymes that have got the different molecular weight, but they show the same function. This is another important term. So this first step is maintained by this enzyme. So how does this phosphorylation gives us information about the goals? First, the hexokinase reaction is converted into non-ionic glucose into an anion that is trapped into the cell, since cells lack transport systems for the phosphorylated sugars. Second one, the otherwise biologically inert glucose becomes activated into a labile form, and it is capable of being further metabolized. And we've got 4 mammalian isozymes of hexokinase are known. We've got types 1 to 4, and with the type 4 isozyme often referred to as glucokinase, and glucokinase is the form of enzyme found in the liver cells. In hepatocytes, we can find this enzyme. So here you can see the saturation curve. The glucokinase is going to reach into a increasing decrease, but hexokinase increase, and then there's a constant level when you increase the blood glucose in this curve. So we can detail explain this figure. We just compare the activities of hexokinase and glucokinase, just the Km values. If you remember, the Km value is the half of Vmax, half of the substrate concentration of Vmax over 2 at half of the Vmax, the substrate concentration at Vmax over 2, that means Km. So this Km value of hexokinase is significantly lower than that of the glucokinase, 10 millimolar concentration, and this difference shows us that non-hepatic tissues which contain hexokinase rapidly and they can be able to efficiently trap blood glucose within their cells by converting it to glucose 6-phosphate. So let's have a break again.